Bienvenue! Welcome to the plant cell. Unlike animal cells, plant cells have a strong protective outer layer called the cell wall. Cell walls are found not only in plants, but in algae, fungi, and nearly all prokaryotes as well. Prokaryotes are one of two types of cells, the other being eukaryotes. Prokaryotes are smaller and less complex than eukaryotes and have no membrane-bound organelles. Therefore, prokaryotes do not have a nucleus. Plants, however, are eukaryotes and do contain a nucleus, which is where we will begin our tour. But first, before passing through the cell membrane, we must go through the cell wall. Most cell walls are made from fibers of carbohydrates and protein, and mostly consist of cellulose, a tough carbohydrate fiber. The cell wall only allows certain substances to pass through, and only through designated entrances and exits. Substances enter and exit the cell through the cell membrane in two ways, through passive transport and active transport. Passive transport occurs when a substance moves from a high concentration to a low concentration. Passive transport does not require cellular energy, whereas active transport occurs when a substance moves from a low concentration to a high concentration. Active transport does require the use of cellular energy, also known as ATP. We are now right inside the cell wall, and in front of you, you will see the cell membrane. The cell membrane is a thin, flexible barrier around the cell. Both the cell membrane and the cell wall protect and support the cell, while at the same time allowing them to interact with their surroundings. The cell membrane is made up of lipids and proteins. To get to the nucleus, we must pass through the cytoplasm, rough ER, and nuclear envelope. The cytoplasm is a clear, jelly-like substance that occupies the space between the nucleus and the cell membrane. The cytoplasm contains many important organelles, including the mitochondria, Golgi apparatus, chloroplasts, the central vacuole, smooth ER, and the free ribosomes. The mitochondria are considered the powerhouses of the cell because they produce a high-energy compound needed by the cell called ATP. The mitochondria and chloroplasts are not part of the endomembrane system and contain their own circular DNA. Mitochondria and chloroplasts both change energy from one form to another. Mitochondria are the sites of cellular respiration, a metabolic process that generates ATP, while chloroplasts, found in plants and algae, are the sites of photosynthesis, converting sunlight to energy for the plants. Vacuoles are sac-like structures that often store materials such as water, salts, proteins, and carbohydrates. The large central vacuole found in plants are larger than those found in animal cells and contain water and nutrients. Pressure in these central vacuoles makes it possible for plants to support heavy structures, such as leaves and flowers. Smaller vacuoles, involved in transporting substances throughout the cell, especially in the endomembrane system, are called vesicles. There are two types of vesicles, transport and secretory. Ribosomes are the bases on which proteins are assembled and are small particles made of RNA. Ribosomes produce proteins according to instructions that come from the nucleus. Free ribosomes are found floating around in the cytoplasm, and bound ribosomes are attached to the endoplasmic reticulum surrounding the nuclear envelope. The endoplasmic reticulum to which the bound ribosomes are attached is called the rough ER. The smooth ER stands separate from the rough ER and does not have ribosomes attached to its surface. The smooth ER synthesizes lipids, metabolizes carbohydrates, detoxifies poison, and stores calcium. The endomembrane system is the process through which proteins are made, modified, and transported around or outside of the cell. The nucleus controls most cell processes, contains the hereditary information of DNA. DNA holds the coded instructions needed for making proteins and other important molecules. The string-like dispersed material visible within the nucleus is called chromatin. Chromatin consists of DNA bound to protein, and it condenses to form chromosomes. DNA unwinds to form RNA. RNA uses the information from DNA to make ribosomes. Right behind us is the nucleolus, where the assembly of ribosomes begins. The nucleolus and chromatin are all enclosed within the nuclear envelope. The nuclear envelope is dotted with thousands of nuclear pores, which allow material to move into and out of the nucleus. The nucleus sends a steady stream of RNA out of the nuclear pores to the rest of the cell. If you'll follow me, we'll continue to the rough endoplasmic reticulum. Right outside of the nuclear membrane, ribosomes create proteins. These proteins are then modified in the rough endoplasmic reticulum surrounding the nuclear envelope. Glycosylation occurs within the rough ER, where sugar is added to the proteins. The unique sugar patterns ID the tissue. Proteins with a different sugar pattern, as from another person, may be rejected by the cell. If we walk to the end of the rough ER, we will come across a transport vesicle, used to transport the modified protein to the Golgi apparatus. The Golgi is the next stop on our tour. Here in the Golgi apparatus, the modified protein can again be modified, after which it is prepared for shipping to the cell membrane or outside of the cell. Another fun fact: the Golgi apparatus consists of flattened membranous sacs called cisternae. The finished protein is then packaged into secretory vesicles for transport to the cell membrane or other locations outside of the cell. So far, I have discussed almost all of the organelles that are unique to plant cells. The organelles and structures that are unique to plant cells and will not be found in animal cells are the cell wall and the chloroplasts. The organelles that are unique to animal cells and were not present during this tour of the plant cell are lysosomes and centrioles. Lysosomes act as the stomach of the animal cell, digesting any waste produced by the cell, while centrioles move and organize the chromosomes found in animal cells. Ladies and gentlemen, the tour is almost over, but first 3 more things. Number 1. All of today's tour, especially the vesicle transportation, has been made possible by the cytoskeleton. The cytoskeleton supports the cell and is a network of protein filaments that helps the cell to maintain its shape. The cytoskeleton is also involved in many forms of cell movement. Vesicles travel along monorails provided by the cytoskeleton. These monorails are made up of many important structures, including microtubules, that are hollow tubes of protein that maintain cell shape and also serve as tracks, along which organelles are moved. Microfilaments, intertwined strands of actin, also make up these monorails. Just a little food for thought. Number 2. Cilia and flagella aid in the motion of cells. Cilia are small hair-like projections found on the edges of cells. There can be up to or more than 100 of these small hair-like projections. Flagella are long whip-like structures, and unlike cilia, there are only 1 to 2 of these structures per cell, rather than 100 or more. Flagella have a snake-like motion, while cilia have a row oar-like motion. Cilia are much shorter than flagella. In conclusion, I'd like to remind you all of one of the founding concepts of biology, called the cell theory. The cell theory states 3 things. Number 1. All living things are composed of cells. Number 2. Cells are the basic units of structure and function in living things. And number 3. New cells are produced from existing cells. I'd like to thank you all for being such a great audience, and I'd like to wish you all a good night.